Namaste. Welcome to our latest episode of Bodh. And I'm very delighted because I have here with us today a guest. I know him now by name, Prakash ji, but I've known him on Twitter as the handle Ugra, UGRA, for a long time. And I've been following that handle with great awe and respect because it's a handle that's been keeping abreast of a lot of latest developments, papers, and for knowledge and findings on archaeology, on genetics, on the matter of Proto-Indo-European linguistics, Aryan invasion theory, migration theory, as you may call it. And I'm very happy to be in conversation today with you, Prakash ji. Namaste. Welcome to our platform.
Namaskara, Amrit. Thank you for inviting me on your show. I was very delighted when you sent across a message stating, can you do something? Because I was already doing something in written format. And I thought this opportunity gives me some, work to jog my mind and to get some questions, let's say, from interested audience on what they expect. So thank you. Thank you again for inviting me. How do we start?
Yeah. So, I think just to set some context, you are also a working professional. I'm also a working professional. And most of the people who will see this episode, who follow either of us on Twitter, are also working people. They're not necessarily people embedded into these fields of academia. So if we can start with, you can just give us a sense of how did you get interested in this matter of ancient Indian history? We can call it many things. We can call it archaeology, genetics, but what interests you? And I assume that these are things that you keep abreast of in your free time, or outside of your work environment, your family background, etc. So how did you get into it? When did you get into it? And why do you stay in it?
Okay. So maybe a brief bit about my background. So I am a chemical engineer. I come from the city of Bangalore, Malleshwaram to be precise. I have a master's degree in industrial management, and these areas, specifically. Before I got into chemical engineering, I was also very good at chemistry, and that's one of the reasons I picked chemical engineering. And genetics, in part, at its base, is about chemistry of life molecules. So there was some, confluence between what I could read a technical paper and what genetics is about. So there was some confluence. So that was, what helped me get in and then made me stay there. When you read, let's say, a paper about aerodynamics, if you do not know enough fluid dynamics and all the associated formulae, you will quickly get bored. But let's say when you know some of these aspects, that helps you stay in. So that's one of the first things, that made me stay in. But even before that, starting from, let's say, 2010 or so, I was very much interested in archaeology. And again, what you call, maybe in different parlance, gateway drug. The gateway drug was IBC symbols, the undeciphered IBC symbols. So that's what got me into archaeology. I used to look at, I used to work in Hyderabad for a brief while, and they have a fantastic British Council library there. And there was this book. When I look at it, it was probably never lent to anyone. It was blank. It was a book of seals by Asko Parpola, the Finnish researcher. He created a corpus, an outstanding photographic, high-quality corpus of all the Indus Valley seals. And before that this was before smartphones, and you couldn't see such stuff in real life, actually, or at least even search for it when you have time. One of the things is time. When you have time, before smartphones, you couldn't just search for something. You had to go somewhere and find the resource, and then you had to get that. So when I saw those IVC seals, I was hooked, actually. So I started reading stuff. I did my master's at IIT Kanpur, and also those surroundings, they have an intellectual debating atmosphere, which is completely different from, let's say, other colleges across India. So even when I was in Kanpur, I was strongly exposed to OIT kind of discussions, OIT versus AIT kind of discussions.
Yeah.
But at that time, it didn't pull me in. But later on, when I started having some free time, and this Asko Parpala's book was actually quite instrumental in getting me into archaeology on a very dedicated way. So that's where it started. 2020 Corona hit us, and then I was already reading and also commenting on these issues, and then that was a really, it was a paradigm change for many of us. We couldn't go outside, we couldn't. I had never worked from home before that, so that was really strange. And even I work in a manufacturing sector, and work from home is completely unheard of. We have to be on the shop floor, so that changed. I started to think, what can I do? I have so much free time now, nothing to do. So what I did was I ordered this Ancestry kit from one of the commercial genotyping companies. And I know you have to spit into a vial, and then you send it away. And then they take it for 3 weeks or a month, and then they come back to you with all details. So I sent it away, and about, let's say, 1 month later, I got details of my haplogroup, which is my paternal haplogroup and my maternal haplogroup. My paternal haplogroup was R1A-Z93. My maternal haplogroup was M30 and one of the subclades. So R1A-Z93 is already, it's
Aryan gene.
It's a meme material on all online discussions. Plus, of course, it's center of what you call as the AIT, OIT, the focus, So we, in my family, I have a pretty good understanding of where we come from, in the past as well. Where our community was settled before the modern era, what would we do? So knowing that I'm R1Z93, and then trying to reconcile what scholars or papers say, was extremely difficult for me to understand. Okay, so does it really mean I come from the steppes? Let's say 3,000 years ago. Does it mean I come from the steppes? It could be true. I'm not saying that it's not true, but that pulls you in. So that pulled me in, Then this commercial company, they have a very good relative finder.
Okay.
So your genetic chromosomes, they have SNPs. So normally with my parents, I would share about 50% of my sequences. And with cousins a little bit less, etc., etc. So this Ancestry service, they found a fourth cousin once removed. So fourth cousin means me and that person has one set of great grandparents. Okay, so I think about 4 generations back, we had one set of grandparents, and once removed means he is my father's generation. And I had several such relatives. So I sent messages to every one of them asking, hey, where are you from? Who are your parents? What is your area? Where do you stay in India, etc., etc. So this person came back, and I talked to my father as well to get some more knowledge, And that, at that time, I realized I know so little of my immediate ancestors. So we do know in a sense, generally, where we come from, what did we do, what was our occupation in the past. But our immediate ancestors, that's like in the next town, who are my ancestors in the next town or relatives in the next town? I don't know. I didn't know. Yeah. So I talked to my father. I got down a list, and then I sent it to him. And then he said, I think your paternal grandmother and me are related. That is he He and
Hmm.
my paternal grandmother are related. They come from the same village on the eastern seacoast of India. I don't know if you have heard of the name Kadalur. That's where the tsunami hit.
Okay. Ji.
So they come from a village there, and it's highly likely that my paternal grandmother and his father were cousins.
Hmm.
Okay, so this is all going on, and then I also googled his name. I found out he is a well-established authority in his field. He has written several books. One of his books I used as a reference material in my undergraduate course.
Right.
So I was like, and I asked him, "Did you write? Are you this person?" And did you? Yes, he said,
Hmm.
"Yes, yes, yes. That's who I am." So it's for me. It was really the power of genetics. At that point, I became a full believer.
Hmm.
So there is a movement in it could be anything. Huh? It could be anything. It could be a health routine. It could be something about personal habits. Someone becomes a full convert. They say, yes, I should go to sleep every day at 8 p.m. and then my health is
Sure.
perfect. I can see that. I can see that it's empirical. Huh? So for me, at that moment, it clicked. Okay, this
Hmm.
this field really has power. It really has power to determine
Yeah.
history. And we continue to talk, and I also found out other relatives who are now in South Africa, in the UK, so in Singapore, in Sri Lanka.
Okay.
That was really nice, actually. Really nice.
Ji. Sure.
um, so at that moment I started looking at a much deeper level into
Hmm.
genetics to understand what is going on. So that is what made me stay in. That was your question. What makes you stay
Right.
in? You could get
Right.
bored
Right.
after some time. But
Yeah. Yeah.
that what made me stay in.
Yeah. I almost had a similar journey myself. For me,
Hmm.
I remember, I think it was maybe 2017, 2016, when that book had come out, The Early Indians, I think by Manu Joseph, if I'm correct,
Yeah,
the author. And in
yeah.
that, he showed the genetic findings that, Saryu Parin, UP Brahmin has this genetics and that genetics. And I looked at that and I was like, oh, well, this confirms it. This means that I did come from the steppe. That's how he put
Hmm.
it in the book. And because
Hmm.
genetics, we are wired to understand it as hard science. I was like,
Hmm.
well, if it shows that, in UP, Saryu Parin Tiwari is from the steppe, then I may as well be from the steppe as well. And I remember at that time, I felt a bit disturbed as well that, oh, I mean, does this really, is everything true? That means all the Aryan invasion theory, all of that
Yeah,
landed back at me at the time that, oh,
yeah,
that means I have a lot of reshuffling to do in my head.
yeah,
And then,
yeah.
of course, I also went into it deeper for a bit to at least understand what the landscape is like. And then, similar to you, 2020, I mean, it's such a weird year, of course, a global disturbance. But
Yeah.
there are so many of us who also were able to get some, let me call it silver lining during such a period
Yeah.
where
Yeah.
we had more time on our hands. So we were able to read things or get into things that maybe we had been, postponing for a long time because work was
Yeah.
going on and all of it was going on. But in
Yeah.
2020, I also had free time, so I started reading into Indian history, etc. Once again, and that is when I did, at least to my level, a bit of a deeper research to get a bit
Hmm.
more acquainted with these things. So,
Yeah.
in that sense, yes, COVID helped me, which is almost a very bad thing to say, understanding the global tragedy, but certainly that's how it panned out for so many of us.
Yeah.
Great. So now, when we talk about archaeology, genetics, and you used a few terms which I just want to tell the audience, we will be talking about AIT, Aryan invasion theory, OIT, out of India theory. These are terms which we'll get to, and we'll expand and unfold them out a bit later.
Hmm.
But as
Hmm.
we understand it, and before we get to genetics, which I'm sure is the meat of the matter here, and you have a lot to talk about, let's start with archaeology. And if you can help us understand that the archaeological landscape in India, we know that archaeology as a field, in a sense, was conceived of and began by the Britishers, by our colonizers who were in India, and they were discovering various things. I mean, we all know the story of how Harappa and Mohenjo-daro were discovered through their bricks were being used for a railway track ballast, etc. But
Yeah,
that is almost the extent of what we know. Most of us know about Harappa, Mohenjo-daro, but very few of us even now actually know of Rakigadi, or even of
mhm,
Kalibangan, of Bhirana,
yeah,
of Sothi, of Siswal, and
yeah,
more recently even Sinauli. And there is
mhm,
still a lot of controversy around Sinauli about the archaeological findings there. Then we also have something like Kizadi in Tamil Nadu, and I apologize for my poor pronunciation if the pronunciation there is wrong. But
yeah.
can you tell us how is the landscape in the last maybe archaeology does move a bit slowly, so maybe in the 5, 10, 15 years, what is the latest landscape of archaeology in India? What is it telling us?
Okay. So I'm trying to compress so many things to give
Yeah,
a very good gist of what is going on. Somewhere in the, let's say, the late 70s and the 80s, B.B. Lal, who
Ji.
is the doyen of Indian archaeologists, he had a paradigm change in his mind.
Hmm.
He started to discard AIT very comprehensively in his speeches, in his papers, in forums across the world. He was one of the champions of the painted graver (PGW) uh, as being the first landing point
Yeah.
of the Aryans when they entered India. So that was his work actually. That was his work. But he started saying, I have second thoughts about this. I don't think that's true anymore. You know,
Hmm.
he started doing that. And that's when, the whole body of archaeologists, and the premier body is now Deccan College in Pune, where Vasant Shinde works. Vasant Shinde is
Ji.
currently, I would say, the most distinguished
Yeah.
practitioner of Indian archaeology. Of course, there are also other people like the Sinoli excavators and also K. Rajan in the south. Uh, but at this point, when BB Lal said this, it's like, let's say, in a political party, someone comes out and says, I don't believe in socialism anymore, and let's say that party has been a socialist party for so many years.
Yeah. Yeah.
So it sent ripples, and
Yeah.
then people started asking questions. How can this be? And they started trying to falsify that. But B.B. Lal showed very meticulously, slowly, that this is the case. One of the things that we know is that painted graver, they were extensive rice cultivators.
Hmm.
So rice cultivators. And in my set of slides, I have something else to show you, which actually convinced almost everyone in the Indian archaeological practice. Why I'm telling you this is it's very tough to find anyone in the Indian archaeological body today that actually actively supports AIT. That's one of the first things that anyone should
Hmm.
know, actually. Um, Hardly anyone. There is absolutely no, when you are a practitioner of something and you have to believe in something. Archaeological is an empirical science. You have to find
Yeah.
something to substantiate what you're saying, and
Yeah.
archaeologists cannot move away from this empirical part of their practice.
Yeah.
This is one of the biggest changes in the landscape, and that coincided actually with the Ayodhya issue.
Hmm.
The change, the change, the mental, the thinking. They came out of the it was Wheeler's theories. Wheeler was a good archaeologist as well. It was Wheeler's theories. They came out of that, and that happened at the same time. Indian liberalization was going on. At the same time, the Ayodhya movement took off. I was just a kid, 10 years old, and now I read about it. At that time, I only knew I had 2 months of holidays in the deep south. In the deep south, it was not such a big issue. But now, when I read all of this, these are tremendous tectonic shifts. The whole paradigm is changing, and at that moment, the countdown for we start questioning, we see everywhere. And then Edwin Bryant, he's an archaeologist. He was doing his thesis. He's young, in the 2000 and somewhere late 90s. He coins the term OIT. He coins the term. We Indians did not coin the term OIT. It was Edwin Bryant who said OIT, out of India. Actually, I see it as a perfect encapsulation of Indian mentality. We said AIT is not true. We said, and then we said we don't care about OIT and all.
Right,
We just said AIT is not true. That was a position of most archaeologists. They never said all Indo-Europeans came out of India. That's basically it's just okay. I'm going to defend, and then I'm just going to stick around the wicket. I'm not going to hit the balls over the crease or something, over the boundary or something. But it was actually Edwin Bryant who first coined the term OIT. And I think Coenraad Elst, who is also someone I greatly respect, he is this Belgian professor,
historian,
who has been deeply involved in all these aspects. He said for a period of, let's say, 5 to 10 years, I think there was growing professional acknowledgement of this reality that we stopped believing it based on evidence. We did not stop believing it based on a political stance. We stopped believing in AIT, the archaeologists, and also general public. And plus, there was no OIT actually. There was nobody saying the Greeks went out of India, etc., etc. So for 5 to 10 years, it was reasonably good. Relations with Indology were reasonably good. They said, "Oh, okay, you have this position. Nice. Can you tell me more?" People are invited. There was Erdosi, who's another practitioner who also published papers about OIT, not an Indian, published papers about OIT. So it was reasonably well. Somewhere in, I think, 2002, 2003, when again the politics was picking up across North India, because I think BJP was ascendant. I think all of this is coming out of a Coenraad Elst article. What I'm saying now, because I was young at that time, I have no clue about. I didn't.
There's so much that we have, of course, learned through his work also.
Yeah, absolutely. Yeah. The Hindu has a magazine called The Frontline. I don't know if you know that. The Frontline published an article accusing, at the same time, there was also a controversy in California about Hindu topics in textbooks. And there was also this attempt, because they were also saying the same thing, that we don't believe that it happened purely based on evidence. Therefore, the other side must also be taught. And then Frontline published this massive, I think it was one edition fully dedicated to this issue, stating that this is Hindutva, basically. That's what they published. And then from then on, it was downward. Relations between someone saying AIT is not true automatically said you are Hindutva. So that's why it started going downhill. So basically, the genesis of this, the politicization, the weaponization of this topic. It's an academic topic. People are perfectly fine to have views based on evidence, but then that's why it started, when the Frontline pushed this matter into the public consciousness, as if it was people trying to get across revisionist history, as if all the evidence was there, and then we were denying the evidence. So that's when it started. It became something different, and I think that shut up a lot of people. It shut because until then, professionals need some amount of what you call as subtlety and civil discourse to operate. To put forward theories and to operate. They need it, and but this actually completely it polarized everyone. And I think that's where it really started going downhill.
Yeah. Yeah.
But I think at this moment, even now, after it happened in 2003, so now we are 22 years, almost a generation ahead of what happened in 2000s, and now we still have the after effects of that. But we have in the last 10 years so many more archaeological discoveries. So the archaeologists, what they did, they said, okay, fine, we will not get into theorizing. We will not get into theorizing. We will not get into OIT-80 discussions. We will not get into that. We will focus on the work. We'll bring out evidence, and we will do what we are supposed to do. So they really put their heads down. Last 10 years, we have a surfeit of new evidence that is actually pointing the other way. So that's going on very well. IIT Gandhinagar, they have this amazing department. There are so many sets of evidence, scattered sets of evidence. They're putting it all together, and they're making it into a theory. So IIT Gandhinagar, they have this amazing department. Some of the stuff I'm going to show on my slides is from them. It might amaze you. I don't know. Maybe you already know that, so I shouldn't underestimate you.
No. I mean, it's for the audience as well.
Yeah, if you've heard of Northern Black Polished Ware.
Ji.
That was a culture that began somewhere in 1300 BCE. It began in the vicinity of Nalanda. I have also tweeted about it. I've written a tweet thread about it. So PGW had a painted grey ware, and then there was black and red ware before that. NBPW was a superior set of vessels. So at the same time, the Iron Age was kicking off in India. So you could actually reduce these vessels in a specific metallurgical furnace, and then they became really strong, and the vessels became really glazed. They were glazed. So they were superior. One of the things is we see across India, NBPW vessels, it would be plates or pots. When they broke, people put them back together and they continue to reuse them. They wouldn't throw them away because it was of such a superior quality, NBPW. Because the supply chain, it was in extreme demand. It was coming out of the Western Ganga Plains. That's not in the East Ganga Plains, the Western Ganga Plains. Let's say towards Bihar, towards what we call as, indeed, towards the lower part of UP. And that's why it started coming out of. Now,
the Magad, what would become Magad eventually, that area, right?
Yes.
Magad Mahajanpat, yeah.
Absolutely. Yeah. What is next is, this IITK Gandhinagar, they put together. So NBPW sites, they start spreading across India. They start spreading across India. They start in this place in the Gangetic Plains, western part, and they start spreading across India. They are the first pan-Indian culture. The first pan-Indian culture. They overrun PGW. So you see over a period of time, they overrun PGW. Painted grey ware is overrun by NBPW. They also reach Maharashtra. They reach the deep south. They are at Kantarodai. Kantarodai is in the northern tip of Sri Lanka. They reach Kantarodai in 900 BC. So they are basically pan-India. By 700 BC, they are pan-India.
So just one thing for also to us to understand, when we say that they are the first pan-India culture or pan-India cultural artifact, what we mean in effect or in practice is actually that because their items, their vessels, their plates, etc., were so durable or of a superior quality, we find them spreading across to larger parts of the country. But does this necessarily mean that prior to this, culture itself was not spreading across the country, or I mean, to what extent can we link the physical artifact of, let us say, some vessels, some plates, some pots, to the larger culture and civilization itself? How do we make that kind of linkage?
Okay. I'm going to bring a modern example. Um, in the modern era, we find a McDonald's in almost every country across the world. On a pure price point and quality perspective, I don't think McDonald's is a superior. It's not a superior food item that provides some specific calorific advantage or price point advantage. No, nothing of that. What it is actually cultural consumption. If someone goes to a McDonald's and eat, it's cultural consumption. It's not the actual the food, the calorific consumption. So that is the imperial spread of American hegemony. Not hegemony. I should not use that word. That's
Sure.
sorry. I should not use that
No, we
word.
get it.
American influence. So NBPW, in the same sense, it's high value elite items. So kingdoms across India start to use it. I do not think that it was superior to everything out there, but the fact that there is something else, there is probably an imperial, what you call as a force, that makes people want to use it. They say, "Wow, that's what somebody was using it in the Gangetic Plains." Now, because you see. Archaeologists can bring forward their findings. Historians have to provide, they bring forward the bones and they bring forward the muscles, but then still you have to put skin on your theory and then you have to put it to the world. I specifically think that the Mahajanapadas already were at their peak in 700 BCE, long before the Buddha. Because there was something coming out of UP and Bihar in 700 BCE, already across the full Indian subcontinent. And that was a cultural impact. If you see the actual historical books, you see they actually lag. And NBPW died out completely by 300 BCE. Which is really
When we
strange.
say died out, what do we mean? That it was replaced by other things, or somehow there was a vacancy or a
It
vacuum?
simply deteriorated. There was no more production of NBPW artifacts. The number of sites using NBPW artifacts, they start going down, and then they completely go to a zero somewhere by 100, 200 AD. They completely go to zero. So, and they are not replaced by anything else. But the peak is in 700 BCE, and then they start going down by 300 BCE. History books, we learn that 300 BCE is when we have all the Gangetic Magad emperors who are actually looking out as they start expanding. There is pan-India extraction going on, and you have Ashoka, Chandragupta, and everyone. Actually, you have a gap between what we see and the archaeological thing. I would actually expect that these both must be harmonized. So we have at least there is this Chandragupta theory, the sheet anchor theory. It might not be as extreme as that, but I think I really think that at least 200 years of we have to push it backwards. At least 200 years, because we see that I put it in a tweet. You can read that. So this era, last 10-20 years, we are seeing archaeologists putting their head down in India, and they are really writing. Probably in the future, in the next 10 years or 20 years, we will see historians not being phased by what Indologists will say. Build up some courage to ask questions and to write at least competing theories. When you have 5 theories, all of them will compete with each other, and you will have the best one come forward. Right now, there is only one that's there. So I think at least that will happen in the coming decade. So that is something that's going on. I don't know if you have seen 3 months ago, TNR, Tamil Nadu Archaeology, they come up with Iron Age objects that's already very early second millennium BCE. Actually, it's quite high. But when you read the report, it's pretty good. Almost everyone supports it. Dilip Chakrabarti, who is a very distinguished reader of archaeology, he's also one of the great historians. He is actually interested in putting out the historical explanation of things, why they happened, what was the environmental reason for that happening, what is the terrain of India when it happened, what were the political reasons. So he's a very good harmonizer of various theories. He supports it. He says that that's true. That's basically what happened, that there was an Iron Age that started much earlier than what we know today. And of course, when the Iron Age is stabilized, you see the number of sites that use iron objects increase, exactly like NBPW. At first, you see in a small nucleus they are using, and then the whole of UP and Bihar use NBPW, and then the whole of India in Gujarat, Maharashtra, Sri Lanka. So that's when you have proper NBPW age. So Iron Age also is similar. It's quite possible that this was a start. You start a flame, it dies down, and 200 years later it starts again. So this new Iron Age finding could be that as well. We don't know, but it really starts showing us that early Indian metallurgists, they were experimenting with new extraction methods. They were trying to make iron, although in isolation, but probably it stabilized by 1500 BC or something. But that's also another thing that happened in the last 5 years.
Right, right. And all of this, what it is actually starting to tell us, because you mentioned how now in recent eras, the archaeologists have started to, at least they've rejected what was the earlier theory about. And the earlier theory was, of course, an invasionist theory. So part of the reason, of course, was also because we haven't found evidence of invasions. But another reason, and please correct me if I'm wrong, another reason was that the archaeologists are finding enough evidence of archaeological, material, civilizational continuities within India. So, for example, you even mentioned rice farming. So now archaeology is starting to tell us that farming was indigenously developed in India. It is starting to tell us that animal husbandry was indigenously developed in India. It is starting to tell us that technologies such as metallurgy technologies, beadworking technologies, ceramic and pottery technologies were also indigenously developed in India. And so that has given to us a different picture from maybe what we might have learned. And for example, you mentioned Dr. B.B. Lalji. He had to go, against what he had himself learned and what he was, the theory he was working on in the earlier era. So that now helps us move to this matter. Let us, before we start getting into invasion or whatever, the matter of population movements. And one of the things that a lot of people often say is that, well, it is obvious that populations have moved across history. So a lot of people actually say this to say, well, why are you against any idea of invasion or migration? Because it's a very natural thing. We all came out of Africa and eventually we all moved to different places. Then we migrated between each other. And this is the story that genetics starts to tell us. So can we move to genetics now? And you have a few slides also. So if you want, you can put them on and we can start there. Or if you want, you can give us some preambles first. I know that there are some terms like ancient ancestral North Indian, ancient ancestral South Indian, Iran, Zagros farmers, some of them are called. Some of them are called ancient hunter-gatherers. What are all of these terms and how do we actually arrive at them? And I am always interested in understanding how scientifically rooted are these terms itself, such as ancient ancestral North Indian, South Indian, or farmer, hunter-gatherer, etc. So where do we
Okay.
start from over here?
Okay. I will put on the slides in a moment, but also to answer that. So those terms ANIS, ASI, Zagros farmers, Iran farmers, Steppe, MLBA, these are all
Yes.
semantic categories. These are all semantic categories. For example, I call something an apple and I call something an orange, right? but actually it's discrete. It's discrete. There is actually a genotype for an apple and for an orange,
Hmm.
and it's physically also I can distinguish them, right? I can do that.
Yeah.
The semantic category that genetics used is based on allele frequency distributions. Is really strange when I say this. Actually, they are a particular statistical artifact.
Hmm.
It doesn't mean that they actually exist in real life.
Okay.
Okay, so like the apple and orange, they are backed up by discrete, distinguishable things. Okay,
Yeah.
at the moment we have semantic categories, but we do not have enough data from all parts of the world. Some of those categories can be discrete, can be really true. Like Iran
Hmm.
farmer can be really true. It can be really true. So,
Hmm.
and I would not say anyone to junk these categories because, because it's not, but we have to keep a healthy skepticism. Um, like I said, genetics is less than population genomics, not genetics. Genetics is actually more than 100 years old. Population, ancient population genomics, admixtures,
Hmm.
these theories are less than 15 years old. Okay, let me give an example. We studied Newtonian dynamics in school. We studied Einsteinian physics also. We haven't studied MOND. MOND is modified Newtonian dynamics. Okay,
Hmm.
so and this field, cosmology and modeling planets and their movements is 200 years old. Okay, it's 200 years old. We are finding out now that the Newton's laws cannot be applied to galaxies, and
Hmm.
this has been gathering steam since the last 40 years, since 1980. Okay, and there are still people are divided, whether they really want to modify Newtonian dynamics. So I'm just giving this example. This is a field that has been well established for over 200 years, and you still have people arguing that this is not possible. We need a third regime apart from Newton's regime, and also at the quantum level, we at the black hole level, sorry, and all we need another regime for galaxies. So this has been happening in the last 3 decades. So nothing is settled, okay, but that's science actually. That
Hmm.
is science, and I urge everyone to understand that genetics is less than 15 years old, and many things that are held as true now will be junked in 5 years.
Hmm.
So that's something we have to keep in mind when we look at it. And it's not like I'm going to say something that's completely bizarre and out of this, but there are published papers that are asking how is this possible, and many of those papers are 1 year old
Yeah.
that are challenging QPADM, for example. So QPADM is the tool that is used to model population histories, and QPADM is a tool that requires very low computational power. You can run it on your laptop. You can upload your genome sequence, and you can find out how much steppy ancestry you have. Anyone can do it in their home. So that
Hmm.
is one of the things why QPADM has become very famous, because it has. I'm going to explain that. But
Sure.
now people are asking if QPADM is correct, and this has been in the last 1 year. So things are moving quickly, and Tony Joseph wrote a book. And if you read, you mentioned Tony Joseph, right, in the beginning, early Indians.
Yeah.
Yeah, he wrote a book, and if my brother-in-law has that book in his bookshelf. I didn't buy it, so I borrowed my brother-in-law's book. But if you go through the book, it has a reverential tone. It is reverential.
Towards the
Yeah, it's
towards the
like,
field of genetics, you mean, right?
yeah, it's reverential towards the conclusions of Rake,
Hmm.
Narasimhan and Lazardis.
Hmm.
It is
Yes.
even
Yes.
reverential actually. But then you don't have to be reverential. Nothing of this is settled. Just because they ran a hypothesis and found something doesn't mean it is true. So this part doesn't reach the average Indian. They really think it is settled. They think it's like an apple and orange scenario. Oh, they found an orange, so it must be an orange. It's not like that. It doesn't have the discrete power. The exact term I'm searching for is empiricism versus rationalism.
Okay.
Empirical truth means you know that it exists. I see the moon. Yes, the moon exists. You know how the universe started. There are multiple competing theories. All of them are possible. Only one of them could be true, or maybe two of them could be true. So that is rationalism. You haven't witnessed that. So most of these things are have a rational nature. I don't deny that there is a steppy ancestry in every one of us, but also how it reached us and what is it are still being subjected to more analysis. And the main thing is it's less than 10 years old, 15 years old.
Yeah.
So that's something that has not reached the average Indian reader. They think it is settled. When I see in Indian airports Tony Joseph's book everywhere,
Yeah.
and they even say it is updated, the latest edition, but actually they are not realizing the fundamental aspects of it.
And I mean, something that really confounds me, I wonder about it, that even when we work from the works of Cavalli Sforza, I think was the scholar, who first tried to find linkages between genetic movements, genetic population movements, and language population movements. And sure, there may be always correlations between them. But even for the simple fact that you and I are talking right now in English, the audience is going to listen to us in English. But none of us here have British genes in us. And there are so many examples of this to tell us that genetics, language, and culture don't always or often move in the same way or in the same patterns. They can always be delinked. There may be
Yeah,
correlations, but those are not necessarily directly causations or linkages.
yeah,
Despite
yeah.
this, why is there so much of a pretense that they are linked? Why do we find so many scholars, thinkers, and I'm not talking about laymen such as myself or the audience who will be listening to this, but there are scholars, linguists, Indologists, geneticists who seem to pretend that just because there is a linguistic movement, that's it. We have found the Aryan gene, for example. You mentioned, Z93, which is called the Aryan gene. So why has this happened, or how does this happen that genes has become a byword for linguistic and cultural movement itself?
See, okay, we have to approach this question from two ends of the spectrum. Um, I'm going to ask you a personal question.
Sure.
Do you think that intelligence is hereditary?
Yes, I think that there are certain dimensions of it or some parts of it that are hereditary. Yes.
Okay, okay. If a researcher said this in today's atmosphere, anywhere in the US or the UK, they would be
Very controversial.
very controversial. Okay,
Yeah.
so it's part of the Overton's window. Right now, the window is at where you just said. The window is at where you just said. Where people really believe that some genetic turnover equals language replacement. So that's today
Okay.
the window is it's in a good way. That they can say that in 10 years it may shift. Okay, so there is really no that's how things go. That's how things go. And it happens with every kind of technology. The window shifts in decades. It shifts in decades. So at this moment, you don't have to fight that. Fighting that, it's like the wind is blowing this way. That's it. You don't have to fight that, and it's pointless to fight that. The wind will shift the other way as well. Just as the question about whether intelligence is heredity linked to heredity and race, for example, it's controversial to even say that it does.
Yeah.
So we'll let that stay. We'll let that
Sure.
stay. But I'll try to explain what is going on in the background.
Hmm. Yeah.
Yeah.
Please go ahead. Yeah.
Yeah. I'm going to present my screen. Can you see it?
It's yeah, we have it.
And it's in slide mode.
Yes.
Okay. Well, so the first thing is, how can ancient DNA, how do they actually propose that this historical event happened or this linguistic event happened? So most of this takes place via the analysis of allele distributions, polymorphism data, and linkage structures. I'm going to talk in detail about what an allele means. Polymorphism. What is a polymorphism? For example, you have carbon as a graphite and also as a diamond. At the base, the material is the same, the element is the same, the physical structuring of graphite and diamond are different. So that is what is called as polymorphism. In the same way, we're going to find polymorphism within the human genome, and that's one of the ways in which um, geneticists figure out which population mixed with other populations. Is that fine so far, Amrit?
Yeah. Yeah. Yeah.
Please stop me and ask questions if you have questions.
I
Because,
yeah.
yeah? Okay. So we're going to look at what an allele is. So allele, basically, it comes from the Greek word for other, also in Latin, I believe. Interestingly, the cognate for other or for allele in Indo-Aryan is aran. If you know the word aranya, which means forest,
Hmm.
or ran for desert. So that's
Hmm.
basically the etymological background of the word allele. So it here in
Okay.
biology, in genetics, it just means the other. Okay.
Hmm.
So what exactly is an allele? So you have these nucleotides, four nucleotides at the heart of DNA: adenine, thymine, guanine, and cytosine. So every gene encodes strands of these four nucleotides. When a single nucleotide is different in the strand between two people or three people, it's called as an allele. So, for example, this is one, let's say this is one whole gene that's being expressed, and this is also the same. At somewhere in the gene, you see a single nucleotide substitution. Here it is adenine, here it's guanine, and here it is thymine for
Hmm.
these three people. Okay, so this is basically what is called as an allele. Sometimes alleles will have an impact, sometimes they will not have any impact. So, for
By
example,
impact, you mean impact on physicality or on the
yes,
biology of a person?
yes, phenotype. It's called phenotype. So
Phenotype. Yeah.
there'll be impact on phenotype. Um, so just like the carbon, diamond, and graphite example, sometimes there is no difference. It's absolutely the same. There's no difference between three people, but sometimes it does. So that's basically you have to keep in mind. So you see sometimes different colored flowers on the same plant or different plants, for
Okay.
example. So that is driven by alleles. So sometimes what happens is when two plants are hybridized, the dominant allele comes out. So the allele drives the color of the flowers. Okay. So this is a gene where this allele is located is called as the locus. So that's just to give the verbiage though, or the exact technical words for it. So everyone has the ABO gene. It's on chromosome 9, and this gene determines the blood group of every individual. Okay. So the ABO gene has three alleles, A, B, and O. And this is the question where you asked, Amrit, what happens to physicality? So in this case, yes. So people have different blood groups, and that's because of alleles. Okay.
Okay.
So when a father and mother they mate and they produce offspring. So you have let's say the father has an A allele and the mother has a B allele, and then within the offspring, so the codominance, the patterns or the recessive patterns determine the blood group of the children. Okay. So this goes on and on and on. So these children they marry other mates, and then their children will carry forward those alleles. So this is a very real example how it impacts physicality. So I think 200 years ago people didn't know about this, and blood transfusions would result in unintended consequences. So we now know exactly down to the genetic level what is going on with the blood groups. So this is an example of an allele that has impact on protein expression. This is another example of alleles. So all of these are genes. On the left, you have this HERC2, OCA2, KITLG, DTB1, SLC24, A5. These are all genes that determine the color of skin. So on the right are European populations, and on the left are West African populations. So these are the genes, and these are the locations of the genes. For example, KITLG is located at chromosome 12. This gene is at chromosome 12, and these also have alleles. So sometimes, unfortunately, sometimes in the West, you see two white parents getting a dark-colored child. That can be because of an allele. It doesn't have to be that it's the same skin color. Sometimes that can happen very naturally. So this is real phenotype expression of a genotype, the skin color. Also eye color, hair color as well are driven by alleles. Yeah. So everyone has 23 pairs of chromosomes, of which 22 are autosomes and one is determining the sex of the offspring. So in these 22 autosomal chromes, we have about 20,000 genes that actually code proteins, and also we have 20,000 genes that do not code proteins. And this number is always going up. They're finding more genes or other sequences that are doing some function within the cell. So the number of genes are always going up. Okay. There are about 15 million SNPs. That's a single nucleotide. There's about 15 million SNP sites. Every individual has 4 to 5 million SNPs. That's alleles. Now, out of these, let's say individual has 4 to 5 million SNPs, ancestry informative markers. Okay. So, for example, when I gave in my DNA, for testing, they said immediately you're from South India. Okay, they didn't call me or ask me, but they said you're from South India because I have some specific alleles that are not even common in North India, but only in South India. Okay, these alleles are highly geographic specific. So that is what is called as an ancestry informative marker. So ancestry informative markers, they are only in, I think the Reich Lab, they published a paper that had 4,000 ancestry informative markers. So different papers use different transcription sets. So you could have, every study could use a different number of AIMs to find out the location of the participant, for example. Clear?
Yeah.
Okay, we come to how history is actually figured out.
sorry, just one maybe also for the audience. ADNA means ancient DNA, right? That's all that means.
Ancient, yeah,
ADNA.
ancient DNA. Yeah, yeah, yeah. That's
Cool. Please
correct.
continue.
Yeah. So let's say in the past 4,000 years ago, we have a source population. Let's say they could be in Sri Lanka.
Hmm.
So they are in Sri Lanka. This source population, they are geographically isolated, and then the allele frequencies in everyone on that island, they become distinct. Okay, the frequencies, they become distinct. Okay. Now, when two source populations combine to form a new population, the allele frequency of the new population will be dependent on the ancestry contribution and the time of mixing of the two source populations.
Hmm.
So, like, let's say the populations in Sri Lanka, they found a way to cross the Park Strait, and then they come over into Tamil Nadu, and then they decide to take mates. The children of those um will now have a different allele frequency,
Hmm.
and that allele frequency will continue, and they will pass it on to their children. And now, for example, in the modern era, we cannot distinguish between uh, we cannot distinguish from autosomal studies for some sites, for some commercial providers, they cannot distinguish between South Indian and Sri Lankan populations.
Okay.
But let's say you have a very good set of ancestry informative markers that are far beyond commercially available, then in theory you could still distinguish who are coming from Sri Lanka and who are coming from South India. But this is the principle. So the frequency set, which is expressed as a matrix, it becomes the identity of a population. The frequencies, the allele frequencies. I'm not sure if it is making sense because I don't hear anything.
No, no, it is. It is. No, no,
It
I'm
is?
following. I'm following.
Okay.
Yeah. Yeah.
Forgot to say something. I took most of this material from a very good paper that is from Shyamalika Gopalan. It's a quite recent paper. It's from 2022, and they also did
Okay.
a study of all the admixture and dating methodologies that have been used, and then they also used it to show a very good example. I'm going to show that now.
Hmm.
So they analyzed about 500 modern individuals from Central America, from Mexico, from Colombia, from Peru, Barbados,
Hmm.
Puerto Rico.
Hmm.
So they analyzed about 500 individuals, and then they checked the allele frequencies of about 700K SNPs.
Okay.
Um, and they found out how different ancestries are distributed. So West
Hmm.
African ancestries, these were introduced to Central America by the slave trades. The
Yeah.
indigenous ancestry are the native Indians in the Americas.
Yeah.
And the European ancestry was introduced by the conquistadors from Spain, Portugal in the 15th and 16th centuries. Yeah, so three distinct
Yeah.
ancestries. Yeah. So now I will show you the difference. So this ancestry is a pie chart. It shows the ancestry overview of all the Puerto Rican samples. Puerto Rican samples.
Hmm.
The majority of them are European, and a small minority are West African and Indigenous American.
Hmm. And
What?
this was their current samples, right? This is not ancient or medieval DNA. It's modern
No,
population samples.
this is modern samples. Yeah,
Yeah.
today.
Yeah.
Yeah. So what this means, this percentage really means that in the population, about 70% of the alleles of everyone is
Hmm.
from an European origin.
European.
That's what they are stating. Okay. This is what
Yeah.
this population level pie chart means. Okay. Now, the
Yeah.
same Puerto Ricans at the bottom, I have another. This is individual level variation. So some individuals are up to 90% European, and some are as low as 30% European. You see?
Mm.
So,
Yeah.
so this is population level pie chart, and this is individual level variation. So even in Mexico, you see there are some people who are barely, um, there are 60% European, and some are 0%
Not even
European
that. Yeah.
over here at this end.
Yeah.
Also in Peruvians is a lot, very less European ancestry in Peruvians. But all of these
Mm.
are showing the distribution of alleles. Okay,
Mm.
it's actually I find it hard to say it. It is what is proposed. It is not. It could also be something else. For example, in this case, though, Shamalika, the authors they selected this example because all of this admixture
Mm.
happened in the last 500 years. Okay,
500 years. Yeah.
that's one of
Yeah.
the reasons they say why they chose this example. It happened in the
Mm.
last 500 years, and before that 500 years, these three ancestries did not interact at all.
Coexist. Yeah. Yeah.
Yes, exactly. And they are very cautious because they are doing basically a worldwide state of the art assessment of genetic methods. So they took
Mm.
the least controversial example. Okay.
Right. Mm.
So these three ancestries they did not interact before in this part of the world. Only in the last
Mm.
500 years they have been interacting.
Yeah.
So in this case, this is true. Now, this is the chromosome set of two individuals, left and right. Okay, one of
Mm.
them is from Colombia and one of them is from Mexico. Okay, from bottom to top are the 22 chromosomes. Okay, from bottom to top, 22 chromosomes. 23rd chromosome is the sex chromosome that determines the sex of the individual, so that is not
Yes.
shown here.
Yeah.
Okay. Now you see, chromosome 1 has the highest number of genes. Chromosome 22 has the lowest number of genes.
Okay.
And on each chromosome, different strands have different ancestries. That these are the alleles. These are the
Mm.
alleles. Okay.
Mm.
So what was red? Red was West African. So West African ancestry is maximized in this Mexican in chromosome 10.
Okay.
Okay.
Yeah.
So we saw that the ABO gene for blood group is on chromosome 9. Okay. So on chromosome 9, there is almost no African ancestry. So this means that this individual's blood group is probably mediated via indigenous American and European, the blood group, because there is
Okay.
almost no African ancestry on chromosome 9.
Mm.
So this is how it looks at the chromosome level. So you have
Mm.
stretches or sequences which are wholly inherited from a different ancestry.
Mm.
So at the population level, you have a different pie chart. At the individual level, you have a different pie chart, and at the chromosome level, it's completely different. Okay.
Mm.
So, for example, you have skin color, hair color, eye color. They are all at different chromosomes. For example, all of those could be from the same ancestry. All those stretches,
Right.
and the
Mm.
individual's skin color, eye color, hair color will be same. And also, this is very good to understand from an Indian perspective because I see
Mm.
several people look at me and they ask if I am, for example, a Sri Lankan or Gujarati sometimes.
Gotcha. Mm.
I don't know why, but they do that. Maybe because it's the way I speak or something, but I also think there is some phenotypic generalization going on, you know,
Mm. Mm.
some phenotypic generalization going on. That also happens
Mm.
for a lot of people.
Sure. Yeah.
So the physiognomy is driven by certain, probably some stretches on these chromosomes. We don't know. There is no GWA study yet. Probably there is. I don't know.
Mm.
So these stretches determine most of how an individual looks.
Mm.
Okay. If that was clear, we'll move on.
Yeah, this is clear.
So now, what is a dogma? So I mean dogma in a neutral sense. I'm not saying it's particularly bad or wrong or something. I mean it in a neutral sense.
Sure.
A dogma is a belief which genetics hold. So what is a dogma here? So admixture graph models. Okay. So admixture graph models is a particular way of stating how two or more populations interacted. I will also show later on why it is different from other models. So they understand that. So admixture graph models are adequate enough to explain population interactions in all kinds of terrains and all kinds of geographies. At the
Okay.
heart of QPADM is a AGS model, admixture graph model. In QPADM, that's the model that everyone uses on their computers to find out how much stepping ancestry that one has. Okay,
All right.
this is one of the dogmas. Prehistory languages can only be transferred by migration and demic diffusion. Okay, so what they're saying is, um, if a language came to India, it can only happen via migration and demic diffusion in the past.
Demic diffusion is when populations are attracted to, let us say, agricultural centers or things like that. Right? What is demic diffusion?
Demic diffusion means two populations merge.
Okay. Okay.
Two populations can come and stay in the same geography, but they will never
Okay.
interact or mate with each other. So you
Then
not
how
only
does
have
the
to
diffusion
migrate,
happen?
yeah, you not only have to migrate, but you also have to mix with the other, with
Ah.
the sex of the opposite community.
Ah.
So that is demic diffusion. So it's not just migration; it's also demic diffusion.
Okay. Okay.
So this is funny, right? Because the problem is, if this doesn't happen, then
Hmm.
geneticists cannot write history.
Yeah.
Because if you see the example that you gave, we have Europeans and English men, Anglo Saxons coming to India. They came to India. They migrated. A few of them diffused demically within into Indian populations, but most of them didn't.
Yeah.
Most of them didn't. So we have a double dogma here. So what they are saying is they not only have to migrate, but they also have to mix. So that's demic diffusion.
Hmm.
Then, of course, using these two principles, they say that we can predict ancestry and history with the ADNA because we have said language can only be transferred by demic diffusion, and we have an admixture graph model that's sufficient to model the population interaction. So
Hmm.
these are the unsaid things that are inside every model. It will never be stated explicitly, but it will be in the model.
Sure.
Nobody will put a fine print saying QPADM has admixture graph, and admixture graph is susceptible or vulnerable to some such specific scenario. So in the paper, it will not be there, but in some other paper, it will be discussed. So people
Hmm.
only reference, Tony Joseph will only reference one paper, he will not reference other paper.
Hmm.
Selective quoting.
Yeah. Yeah. That's it.
Okay, maybe just for a short break.
Sure.
Any questions so far?
No, no. Of course, I'm following you slide by slide and commenting where I had some questions. But yeah, we can continue.
Because the allelic distributions are a bit hard to get, the allelic
Hmm.
frequencies. So that was very clear.
Yeah.
Okay, okay. I'm just going to restart. If you see my screen, please tell me.
Yeah. It's just loading. Just a second.
Yeah, you can see it.
Not yet. It's just loading.
No?
Yeah.
Okay. So let's say there is a population that's isolated, like we said in Sri Lanka, it's isolated. They are not interacting with anyone else. Okay. Allele frequencies within the population will still change, even
Okay.
though they are not interacting with any other population. Okay.
Okay.
They will still continue to change.
Hmm.
So even though they are not interacting, and this change is governed by the Hardy-Weinberg Equilibrium. So Weinberg was a German. Hardy is from the UK. This is G.S. Hardy, who collaborated with Ramanujan, the same mathematician.
Okay. Okay.
It's the same mathematician.
Okay.
So in the early stages of genetics, all these alleles. So there is an allele for blue eyes. There is an allele
Hmm.
for brown eyes. There is an allele for black eyes. So somebody asked the question: is it not possible that over a period of time, everyone gets black eyes or blue
Hmm.
or green? One allele starts to dominate, and then everyone in the population gets the same color of eyes.
Hmm.
So Hardy and Weinberg, they wrote a mathematical proof, and they proved that allele frequencies will remain constant within a population. Allele and genotype frequencies, they will remain constant within a population. Between individuals, they will vary, but overall in the population, if the population has 100 people, the same frequency of green eyes, blue eyes, and black eyes will be there. That frequency will remain constant. So that's the Hardy-Weinberg equilibrium. It's
Hmm.
important to understand this because that has a lot of implications for history, and it's one of the underappreciated things that is not paid enough attention. For example, in many of the models, that's why we were looking at it. Um, so, so there are some assumptions for this equilibrium, and again, this
Hmm.
equilibrium is theoretical. It never happens in the real world, the equilibrium. But when it happens, these 5 criteria must be met. Okay.
Hmm.
So there is no selection. I will give an example what I mean by selection. Selection means some alleles
Hmm.
are beneficial for the survival of the organism. Some alleles. Okay. In that case, what will happen? Only organisms that have that specific allele will survive.
Hmm.
And over a period of time, they will be everywhere. So in
Yeah.
this case, for the equilibrium, we say no selection is taking place. We
Right.
also say that no mutation is taking place. So something has happened, and there is a new allele suddenly. No mutation is
Hmm.
taking place. No migration. So the populations are still separated. Population is very large, and then everyone picks a spouse or a mate randomly. They don't
Hmm.
they don't pick specifically.
So
Okay.
that's kind of it's the flip side of no selection, right? If there is no selection happening, then there will be random mating, and conversely, if there is selection happening, then it wouldn't be random. Isn't that the case?
No, selection in this case is actually referring to evolutionary pressure. It's not the same. I will show it
Acha,
with an
acha. Okay.
example. I will show it with an example. Random mating really means that let's say in this, there are 3 specimens here, right? So they are randomly mating with each other. They are not
Hmm.
picking something specifically. So that's what means
Sure.
random mating. So this is the next example. What happens, what we call as drift. Okay. This example I'm showing, because this can happen in case of selection, and also it can happen in case of drift. So I'm showing this example. So we have this whole population. and then there is a small population that gets isolated in this whole population for some reason. They get isolated. Okay.
mm,
Over a period of 5 generations, you see that the green allele is completely lost.
mm,
Okay. This is
yeah,
called fixation, and it can happen due to selection or due to drift.
okay,
Okay. After 5 generations, one allele is completely lost in a small part of
mm,
the population because they got separated from everyone else.
mm,
Okay, so this is called drift. Just
yeah,
keep it in mind. Now, so we know that most tigers have orange skin, right?
mm,
And that orange skin is because of a gene and specific allele that is present in tigers. And there are also other
okay,
alleles for other skin colors, but we see most tigers are orange color.
mm,
Okay, so what happened in the past? I think millions of years ago, what happened was one tiger first got this allele of orange skin.
mm,
Okay,
mm,
all deers, antelopes, herbivorous animals, they are dichromats. Dichromats means they can see only two colors. They cannot see red, they can see only blue and green.
mm,
Humans are trichromat, so we can see three colors: red, blue, and green.
mm,
Okay, so we can see three colors. Now, this one tiger that got this specific allele somewhere in the distant past, it became
mm,
very successful because deers and everyone else they couldn't see this specific tiger.
mm,
So this tiger, what happened? It became very successful at hunting, and it passed on
yeah,
its genes and its alleles to its offspring, who also became
mm,
very successful because the deer could not see it.
mm,
And in now, most of the tigers are orange. They have orange skin.
mm,
So this is an example of selection. Selection by
okay,
the environment, not
by the
selection by
environment,
the
yeah,
tiger, but selection by the environment.
yeah,
So the environment said, okay, you got orange skin, the deer can't see you, so you have a higher chance of hunting successfully.
mm,
So this is what I mean by selection.
okay,
Okay?
yep,
Yeah? Okay.
yes,
This is drift. Okay? This example is for drift. Okay? The same thing that I
mm,
showed, 5 generations. We have equal alleles of the blue and red alleles. Okay, here in this side. Now, inside these balls, there are dots. These are the number of children each individual will have. So this individual will have one children. This will have two. This will have one, one, one. And you see the blue balls, they have more children
mm,
on average. This one has three, this one has five, this one
mm,
has two, etc., etc.
mm,
Because they have more children, after one generation, there are more blue dots of the allele.
yeah,
The blue, yeah, red has gone down.
mm,
Third, it becomes more. Fourth, it becomes
yeah,
more. Finally, fifth, there are no red dots anymore.
got it,
This is called drift. So the red allele got exterminated or extinct from the population
mm,
because of a decision that it had no power over. At least in the orange skin, the allele had power because it provided the orange skin to the predator and then it got selected by the environment.
mm,
This is the other way. It's drift. Drifting means it has no power. The organism has no power due to
mm,
some reasons, maybe some choices made by these individuals. Their alleles vanish in the population.
mm,
This is called as drift. Please keep this in mind. Okay.
okay, yeah,
The reason I say this is it's important to come when we come to the discussion about steppe and Indian populations.
sure,
Okay. So drift and selection are two important things that are driving allele distributions. Now, both fixation, drift, and selection are driven by the size of the population. Okay,
mm,
there are three examples here. First one, population, there are 20 individuals. Second one, population, there are 200. Third one, population, there are 2000 individuals. The first population that has only 20 individuals, at the end of 50 generations, some alleles will be exterminated or become extinct due to drift. Some alleles will become fixed at 1 due to selection. Okay. In the population n is 200, the same effect will happen, but it will not become 100%. It will not no allele will become fully extinct or fully fixed. And the population is very high, drift and selection will not happen. Because the population is so huge and mating is so high between random mating, that the risk that some allele becomes extinct or some allele becomes fixed at 1, for everyone has the same, that will never happen. And the population is very high. Okay. So now do you understand why this is important in the context of steppe in India?
I hope you detail that. What does confounder mean? It says here, this is the most important confounder for steppe Indian inflections in antiquity. What does that word mean? Confounder?
Yeah, I'm coming to that.
Confounder.
Yeah, I'm coming to that. So this is an example of a founder effect. So there is an original population that they have, let's say there are 5 alleles in this original population. And this is a bottleneck event. This bottleneck event can be an earthquake, it could be fire, it could be drought, it could be disease like plague. So after this bottleneck event, only a few individuals survive. Okay, in this set of individuals, not all the alleles are there. Only 2 types of alleles are there: green and orange. This is called a founder effect.
Okay.
This is called a founder effect. So basically, we take out a few individuals from a big population and then we put it, and then they reproduce and they produce another population. What will happen is the number of alleles in this surviving population will be very less compared to the original population. So that is the founder effect.
Okay.
So we saw drift, we saw selection, we saw the effect of population size, and we saw the founder effect. So these are actual mechanisms that are working every day inside our bodies. I'm going to come. We talked about QPADM. So QPADM is a tool that is widely used today to basically to analyze the allele frequency correlations. They are created using F statistics, and they estimate ancestry proportion from relative differences of alleles. Okay. So this QPADM tool has some assumptions. Okay. If the assumptions are violated, it will not be true. So today somebody says, in general, Punjabis have higher steppe ancestry. Okay, that's based on a QPADM method. Okay, but if some of the assumptions were violated, that would not be true. So I'm talking about methodological errors. Okay, so we're going to talk about methodological errors for QPADM. Vagheesh Narasimhan, I think you've heard about this paper.
Yeah, this is a famous paper. Yeah.
This is a famous paper, very famous. It's a pioneering paper. First time we have ADNA from the Indian subcontinent, and then we found out what is that relationship to the Iranian equivalent. So it's really a pioneering and path-breaking paper, and they were working under extreme constraints. So it's really a good paper in terms of conclusions. Okay, so in terms of data, in terms of data, they're really a good paper.
Data, yeah.
But of course, in the last 5 years, we have found out that they are archaeologically insufficient, methodologically dubious. The conclusions do not match the data, what they have in the paper itself. So So this is the paper that said steppe populations arrived in India between 2000 and 1500 BCE. This is the paper. This is the paper that also is the basis for Tony Joseph's book.
Yeah.
Yeah. One of the first things is that the data from the paper, these are all the points where they extracted data, ADN data. This is the broad Indian zone, and even below this where they don't have data. They have data for Rakhigarhi, that's somewhere here, and of course they have data for Swat Valley in Pakistan.
Yeah. Shari Sokta. Yeah.
Yeah, Shari Sokta is somewhere here, but they do not have data for this area. Okay.
Yeah.
This is important because I believe that populations here are not represented. Populations interactions with these outlying areas are not represented. We have no idea of what was going on in this area in the same period that they claim that steppe populations were entering India.
Yeah.
At least one conclusion was good. They found out that Indians and Iranians are linked deeply in time, maybe greater than 10,000 BCE, because this Neolithic ancestry that is common to IVC and also modern Indians is the same ancestry in Iranian farmers. So this is a very good finding, and they couldn't sweep it under the carpet, so they put it out. It's a central infographic in the paper. But the problem is they didn't interpret it. They have the same ancestry, so they must be talking one language, right? IVC and Iranian farmers and herders. So they didn't link it to Indo-European languages. The fact that Indo-Aryan and Iranian languages are deeply linked, and they didn't link it to this. So it's they fell short of interpreting it.
There is also some time issues, right? So for example, the Indo-European linguistic split or timelines, they are relatively more recent compared to this split. So, for example, the split between the Indian and the Iranian ancestry goes back around 10,000 years, 7,000 years ago. Whereas the linguistic timelines place it maybe some 4,000 or 5,000 years at the most. Is that also a reason why they didn't make that linkage here?
So Narasimhan and the team of authors were fully operating under the steppe model. So they already decided that steppe is the source for Indo-Aryan languages. So it was circular argument.
Yeah, okay.
So they already decided. They said Rakhigarhi woman doesn't have steppe ancestry, therefore IVC could not have spoken Indo-European languages.
Yeah.
So it was circular argument. They didn't see that. See, these Iranians and Indus Valley have the same ancestry. So what is going on? So they didn't interpret that. But anyway, it has good data. For a paper that was so early, it had good data and good conclusions for some. Good data. So this is where I'm showing some archaeological statements. So this is from Berger et al. It is a 2023 paper, 4 years later than Narasimhan. So this is the time from 2300 BCE to 1000 BCE. So this is BMAC, this is Andronovo. So Andronovo are the steppe cultures. They did not extend beyond South Tajikistan and Central and East Uzbekistan. Okay, so they came all the way till 1400 BCE to the south of Tajikistan and Central Asia. Beyond this, they didn't go. And we must keep in mind, Punjab is still 1,500 kilometers away from Uzbekistan.
Yeah.
So this is a pretty good paper, Berger et al. So Narasimhan thought that steppe ancestry came in that era, but archaeology says something else. They said there is no steppe culture beyond Central Asia after 1400 BCE. In fact, they just deteriorated and went away to nothing. And this is another paper from Grigoriev, 2021, a Russian researcher, he also more or less says the same thing. So this underlined part are the Andronovo cultures. They don't go anywhere, like they don't come into painted graveware or anywhere. They stop in Central Asia. So it's not just one paper, more than one paper says the same thing.
Attesting that, yeah.
Yeah. So that was archaeology, but I'm going back to QPADM. What is the shortcoming of QPADM? So so populations, how do they interact? So there are several models in genomics. One of the most famous model is the SSL. SSL means stepping stone landscape. So it's number C on the left corner bottom. So if you go to a garden or or a park, and there is a pond there, and you have stepping stones in the water to cross the pond, for example. Have you seen such ponds?
Sure, yeah.
Yeah. So that's the stepping stone model, stepping stone landscape. So these are the stones, or these are the populations where people are living. So for example, alleles cannot be transferred from this stone to this stone. It can only be mediated via other populations that are in the way. Okay? This is actually central. It supports what archaeology tells us. Andronovo, Sintashta cultures, they died away in Central Asia. They didn't progress beyond that. But it's quite possible that their alleles, they were mediated via other dames in the path. For example, Shakas
Okay.
or Kushanas, for example.
Hmm.
So stepping stone landscape is a completely different kind of model. So PADM uses admixture. It just, if you give two sources like Sintashta and Iranian Neolithic, and it will say, yeah, these two combine to form modern Indians, because it doesn't know stepping stone landscapes.
Doesn't know.
Yeah, it cannot work,
So,
because stepping stone
in
landscape.
just a humorous, in just a humorous sense, like we have Aryan invasion theory, migration theory, trickling in theory. Now we can
Yeah,
also have Aryan stepping stone theory.
yeah, yeah. Yeah. The reason is QPADM. I told you before, it's very easy to compute. It requires very less computational power. You can run it on your laptop. To run the stepping stone landscape models, you need a lot more computational power. It's much more complex.
Okay.
It's much more complex. Recently, I have also tweeted it about it. We have found AHG, Andamanese hunter-gatherer ancestry in Iran in the copper age, Chalcolithic. That's 4000 BCE. So I asked if someone thinks that Onge tribals, they took their canoes and then they paddled across the Bay of Bengal,
Right.
Arabian
Yeah.
Sea, and landed in Iran. No, it's a stepping stone landscape. So the alleles, they were transferred to another deme, and then it reached Iran.
Sure.
So that's one of the problems of QPADM, that it fails, and this is actually coming from a paper, Flegontova et al. This paper is 1 month old. This paper is 1 month old. Flegontova, they have tested all the scenarios where QPADM fails. All these scenarios. So for example, so what you see here, there is S1, S2, and T. So these are the source populations and these are the target populations. And these are the out groups or reference populations that are outside. So there are 5 complex scenarios. And under all these 5 complex scenarios, QPADM does not deliver a good result.
Okay.
So this is now a paper actually. It's true that there is stepping ancestry in modern Indians, but how it arrived there, it cannot be accurately predicted by QPADM. Because archaeologically, we already know something. We already know something that they didn't extend beyond Central Asia. And of course, I don't think alleles can fly through the air like pollen and reach individuals. They have to come via reproductive mates. Another data point, Sintashta culture. This is what everyone says was Indo-Iranian. At their peak, they had less than 10,000 individuals. Max was 10,000 individuals, the population size. Okay. IVC, they had 2 million individuals. Okay. So 10,000 versus 2 million, I think you're looking at somewhere approximately a 200 time magnitude, 200 time difference. So that's what is one order of magnitude higher, the population size. Now, if you go back to the fundamentals and you think about what we just discussed about allele fixation and drift in small versus large populations.
Yeah.
So, okay. So IVC, also the Indian subcontinent, not so IVC is only a part of India, right? We also have the huge Indian subcontinent beyond MP towards the east into the Gangetic Plains and to the south, Deccan Plateau. It's not just IVC, huh?
Yeah.
So bigger populations, they tend to retain their allelic distributions for a long time. Alleles will not go to zero; they will not become extinct. Okay. Whereas in a smaller population, it's much more likely that they become extinct. So the odds or the probability is that populations from the mainland, which is IVC, Iranian plateau, they went into the steppe, and then some of the alleles disappeared is very high, forming the steppe populations. The reverse is not true, cannot be true. And if you see, we have only one individual's data from the IVC. Only
So,
one
I mean,
individual.
just to see if I'm understanding this correct,
Yeah.
is this kind of implying an out of India model even from the genetic data? The way that you say, for example, that it is possible that there were some alleles that went from India to the steppe, only some of them stayed, the others disappeared.
The
Whereas the
alleles.
converse might not, yeah, whereas the converse might not have been that likely or that possible. Can this be understood as a kind of out of India model even for genetic markers? I mean, leaving the linguistic arguments aside, I mean, I'm talking just from the genetic point
Yeah,
of view.
purely on a genomic model. So these demes, I have these stepping stone
Yeah.
demes, right? So the steppe is small n. This n is huge population size. Huge population size. I mean, it's not the same size as the steppe size, deme size. This one is
Yeah.
huge. It's
Yeah.
much more likelier that the arrows going outward from this are much more likelier than small arrows reaching here. Of course, I'm not saying that nothing reached it. Of course, they would have reached.
Yeah. Yeah.
But the computational power to resolve such models are not with us currently. Currently, we have one of a methodology called QPADM, whose model is very simple. And in that simple model, we get an interquartile range of 18% average for steppe ancestry in Indians. So this is the best for the into India theory. For out of India, if we have to compute with the other model, the stepping stone landscape model, that number will probably come down even more. And we already see Asi or Andamanese hunter-gatherer distributions in Iran in 4000 BC.
Yeah.
We already see that. I don't know about out of India, but I definitely think out of Iran is also a possibility.
Yes.
No,
Yeah.
to give you have to be fair enough. It
Yeah.
does not
Yeah.
mean it has to come out of India all the time.
Sure.
It's quite possible that steppe populations also came out of Iran. Iran also had a very high population.
Yeah. Yeah. Please
So,
continue.
sorry, yeah,
Yeah. Please continue.
yeah. Even within the QPADM world, I'm not talking about stepping stone landscapes, but also within the QPADM world, the consensus on a steppe incursion is breaking down. The Amjadi paper that came out 3 months ago, they are saying that there is no steppe ancestry on the Iranian plateau. They are saying there is genetic continuity from the Chalcolithic, that's the Copper Age, to the Sassanid Empire, which is like 300 CE, 300 AD. Also, we see in the data there are no R1As, no R1As
Big.
data.
Yeah.
So even within the QPADM world, things are breaking down. So maybe what else we have? Yeah, this is something else. I'm going to stop sharing. Maybe I think the average person has to know that nothing is set in stone. There is a single methodology that actually cannot handle complex scenarios. That cannot handle a stepping stone landscape. Plus, we have to keep in mind there is no data for the whole of India. There is only one woman from the Bronze Age, data from one woman from the Bronze Age. And because India was such a huge population dame, even in 2000 BCE, there were 2 million individuals. It's highly likely that the steppe ancestry was already in India even before 3000 BCE. So these allelic distributions, we should not call it steppe ancestry because that's how it's being called. When we say this ancestry was in India in 3000 BC, they will say, oh, then also it came from steppe. No, we're talking about an allelic distribution. We don't have to call it steppe. We can call it anything else. If it was already in India in 3000 BC, that means it went out of India. That's what the power of semantics has. So you have this allelic distribution, and then you call it steppe ancestry. Then you actually you're running away with the argument by calling it steppe ancestry. It's just a distribution.
Very fair. Yeah.
So we don't have data for India. When we get enough data for India, then it's probably going to change. We're going to get more harder to compute SSL models in the future. QPADM, already QPADM in Iran. With QPADM in Iran, there is no steppe. There are no R1s.
Even in Mycenaean Greeks, I think they haven't found R1s. Is that correct?
Mycenaeans, they were already having Iranian Neolithic, the same ancestry as IVC. So
and R1A was. Yeah, please go.
Yeah, R1A was not there probably. Yeah.
Cool. So I think this gives us a very good landscape even of the genetic matter. There is, of course, now the final part of it, which would be to also understand this from the in context of linguistics, the Proto-Indo-European homeland issue, Aryan invasion, migration theory, etc. But our conversation has now run almost 2 hours. Would you suggest that maybe we can break this into 2 parts and have another conversation maybe next weekend or at the next time of your availability?
Sure.
And then in part 2, we can discuss the remainder of this. Is that good for you?
Absolutely. This was long enough. And my laptop is also giving up. It has no charge. But definitely we can take this up in a second part. But also I want to show you one last slide before we break up.
Please. Yeah.
That's the NBPW expansion. Let me know when you can see it.
Yeah, it's visible.
Yeah. So this is from an encyclopedia, and it is slightly old because the dates are older here. They are not been updated.
Okay.
So these dark green zone are the NBPW zones.
Okay.
NBPW starts out of this area, somewhere close to Magad, let's say, and then it expands all over India. It goes to Somnath, goes into Maharashtra, then goes south, deep south, goes to Odisha, goes into Takshila, and then this is PGW. PGW just it ends here. Actually, this is the widest extent of PGW. And NBPW starts from the east and goes into the north, goes into the west, and goes into the south. And we see that NBPW starts even earlier, probably by 1000 BCE. So there's really no cultural, archaeological culture that is coming down this corridor into India in that time period. Whatever is there is going
Okay.
out.
Yeah. Yeah. Yeah.
Yeah.
And do we already know that NBP is also not found even further from this, or it's possible that maybe tomorrow there's a new archaeological finding, let's say in Bactria or somewhere in Iran, and we could find NBP there also. Is that possibility open, or we already know this is the route of it?
Um, I haven't seen data for finds in Iran, but probably
Okay,
if it's there, probably it will be mentioned in a paper.
sure. Okay, great, Prakashji. Thank you so much for this discussion.
Yeah. Thank you. It's good speaking
Thank
with
you,
you.
and we'll have part two of this maybe next weekend or at your availability.
Yeah, sure. All right.
Thank you very much. Namaste.
Thank you, Amrit. Thank you.